Our muscles are made of bundles of muscle fibers. Each muscle fiber is made of many myofibrils, and each myofibril is made of many sarcomeres. The sarcomere is made of alternating thick and thin filaments. These filaments are what allow our muscles to contract and relax. Let's take a look. The thick filaments are myosin, and the thin filaments are actin. When these two slide along each other, the entire sarcomere and muscle contracts. The myosin head attaches to a binding site on the actin filament. The resulting cross bridge moves towards the center of the sarcomere. This is how the sarcomere shortens. The two filaments then release once they are in their new position. This allows the next myosin head to attach to the next binding site on the actin filament, and the process repeats, further shortening the sarcomere and contracting the muscle. So how is magnesium involved? In order to attach to the actin filament, the myosin head needs energy. It gets this energy when the ATP molecule it is attached to is converted into an ADP molecule, adenosine triphosphate into adenosine diphosphate. After the myosin head binds to the actin filament and the actin filament slides further down, the newly converted ADP molecule is released from the myosin head. Now another ATP molecule binds to the myosin head to release it from the actin filament. This allows the process to repeat with the next myosin head and binding site. The critical factor is that magnesium is a part of both the ATP and ADP molecules upon which this entire process depends. Calcium's role in contraction helps explain how magnesium is also necessary for the relaxation phase of this contraction. See, the actin filament is surrounded by two proteins called troponin and tropomyosin. Tropomyosin in its resting position blocks the binding site on the actin filament. When calcium attaches to troponin, it forces the tropomyosin to move out of the way, no longer blocking the actin binding site, and thus allowing the myosin head to bind and start the contraction. The critical factor here is that once the actin and myosin binding behavior results in the full contraction of the sarcomere, magnesium is what removes the calcium from the environment, thus allowing the muscle to fully relax.